G'day and welcome to MELMAG Live. I'm Emily Coolidge and this is MELMAG's live stream interview show. Today we're talking to Grace Boxshall, a mycologist and science communicator responsible for the fungi site, Facebook and Instagram pages, among a lot of other things. She's just completed her research into, now I'm going to probably have to be corrected in a tick, for the name of this fungi. It's the yellow stainer commonly, but I'm afraid I've lost it, Grace. Grace, this is a wonderful interview, and it's a wonderful start from my part. What's the name of the mushrooms, that you've been doing your research on?
Agaricus xanthodermis.
Thank you. And you've just had your research published into the yellow stainer mushroom variety, which is exciting, and you're here to talk to us in part about that today. But if we can start off, please. You are a mycologist. Might be a new term to some people viewing. What does a mycologist do?
Well, a mycologist is basically just a mushroom or fungi scientist. So it's just someone who researches fungi. In my case, I look at mushroom evolution, as well as a few other things. So the research that you talked about had some chemistry thrown in for fun. I'm not a chemist, though. That was a lot of hard work to make that look decent. But yeah, mycologist, mushroom scientist.
Very cool. Now, what was it? You had some interesting findings into this type of mushroom that you were researching.
Yeah, totally. So the mushroom that I was researching looks a lot like edible mushrooms. Particularly, it looks like button mushrooms or portobello, anything like that you would find at the supermarket. And it looks like them because it's very closely related to them. But the trick is that they're sometimes poisonous and sometimes not, which can cause a lot of trouble. So when they do cause symptoms, they cause gastrointestinal irritation, vomiting, diarrhea, stomach cramps, just generally a very bad time. And so my research was trying to work out why that variation occurred. And we found out that the concentration of the poisonous chemical in these mushrooms, so phenol is the chemical, we found that varied a lot in concentration. So some mushrooms within the same species, some mushrooms were more poisonous than others, and it depended on a range of different things like the substrate they're growing on, the developmental age of the mushroom, part of the mushroom that you eat, all sorts of things like that.
So we should actually start us off with a huge qualifier that this is absolutely not advice if you're interested in going out looking, foraging for edible mushrooms. Unless you actually really know what you're doing, that's quite a dangerous thing to do. What kind of... These are just regular mushrooms that people find on the side of the street. You on Fungi site, I saw you posting a couple of videos of you just sitting on a nature strip with some very regular looking mushrooms that I think I've walked past hundreds of times. These are mushrooms that people are used to seeing about the place.
Yeah, no, they love nature strips. They love nature strips, parks, gardens, football fields. Yeah, if you've found a mushroom that looks just like your normal edible mushroom growing on the side of the road, chances are you've found Agaricus xanthodermis or the yellow stainer, so that poisonous species.
Now, they get that yellow stainer name for a pretty obvious reason, isn't it?
I mean, it's obvious once you know that it does it. I didn't know about these mushrooms until I started doing a master's on them. And I saw photos of this yellow staining, and I still didn't get it until I went out into the field, found some of these mushrooms and cut them in half. Basically, they're called yellow stainers because when you cut, scratch, or bruise the flesh of the mushroom, it turns bright yellow, like really, really vivid, bright yellow.
Usually. I think that was part of your research I think you noticed, was it waterlogging or something along those lines that made them not quite so obviously yellow staining?
Yeah, so they're absolute trolls, these mushrooms. They like to trick us. So the ways that we identify the species of mushroom is, yes, it stains yellow if you cut, scratch it or bruise it, unless it's been raining recently, in which case it'll probably be waterlogged, so it won't stain.
And
You
that's
can
also when it's been raining is when you most expect a mushroom to appear as well, isn't it?
yeah, so cheeky. But then also, like, you can identify them a bit by smell. So if it smells really bad, like chemicals, phenol, if you're familiar with smelling phenol, printer's ink, wheelie bin cleaner, any sort of cleaning, chemically unpleasant smell. But the trick with that is that only about 40% of the population can smell phenol. And also, if it's cold weather, you won't smell it.
That is really hard to identify. And I think, what was it? So one key part to your research here was setting up Fungicide, which you, along with someone else, you founded and maintain along with someone else. And that was as an I believe, a citizen science outreach. You asked people to let you know when they saw mushrooms similar to the ones that you were looking for on their nature strip or on their walk or in the park, and let you know so you could go and check them out.
Yep. Yeah, no, that's exactly it. So Fungicide, I started it mostly myself, but with my supervisor, Teresa, also on board. And really, it was just initially to help me with my fieldwork. Mushrooms are very hard to predict. You don't know exactly when or where they're going to pop up. You know, roughly autumn, but apart from that, it's a bit hard to predict. So my way of finding where they were going to be and when was quite literally getting eyes on the ground. People go on about their business to let me know if they saw my mushrooms so that I could then go out and collect them.
And it was a mix. How much did that response contribute to your research?
It was huge. So I think I worked out that about 39% of the collections I made for my master's were informed by Fungicide, which it doesn't sound like a lot, but that is actually quite a bit. So the majority of my collections came from herbarium material, so things that have already been dried and preserved and tucked away somewhere. But then 39%, so nearly 40% came from people telling me that they'd spotted my mushrooms nearby.
You mentioned dry samples there, and there's a lot more to researching mushrooms than just picking them from the side of the street. What did an average day in your research kind of look like? Like, yeah, I guess how did you go about this project?
Well, that project in particular was a bit different for me from what I do now. But sort of average day in the life varies a huge amount depending on the season. So in mushroom season, it is all hands on deck, running around frantically. So what I'll do during mushroom season is I'll spend some time getting my field kit prepped and ready to go. So that for me is a backpack full of, I've got a ruler, paper bags, a bread and butter knife like this one that of course I have sitting on my desk. I use that just to uproot the mushrooms. Pens, pencils, plastic tubes with like a sealable lid, permanent markers, all sorts of stuff. Getting all of that ready to go, and then basically being able to go out into the field at the drop of a hat, and travel around collecting mushrooms. And I can spend one mushroom collection is usually about 30 minutes to 40 minutes of me sitting on the ground with the mushroom, recording stuff like the Latin long, the surrounding habitat, whether it's urban or native bush or something like that, making lots and lots and lots of notes about the mushrooms I find. Then, cutting them up, doing all sorts of things, then taking them back to the lab and spending another 45 minutes making more notes about that mushroom. I also take a sample, put that aside for DNA. And for my master's, when I was looking at these poisonous mushrooms, I was also taking a mushroom, putting it in the freezer so I could use it for chemical analyses. I also, on top of that, then everything else that I haven't used for DNA or chemistry gets cut into sort of 1 centimeter thick-ish quantities and dehydrated. That's dehydrated for between about a day or so, and then parceled up. Everything's labeled very, very well so I can find it and know exactly what's what. That's going to be my voucher specimen or herbarium specimen, it will be stored forever in a herbarium so that someone in the future can come back to it and either double check my science or use my stuff for future science. So kind of cool. That's just field season, so I'll be doing that sort of day in, day out and trying to curate photos because I take way too many photos of everything in the field because once they're dry, they just look brown. So I try and capture all sorts of things like colours, shapes, structure, all of that as much as I can in the field.
And all these details with location of where the mushroom was and the kind of conditions they're in, and roughly where, if people are wanting to get their mushroom, like if someone is trying to ask you what a certain mushroom is or something like that, that's important information for them to include as well. Obviously, maybe don't handle the mushroom if you don't know what it is and you're not sure on how to, best clean your hands as a result of potentially handling a poisonous mushroom. But that's the kind of stuff you look for from anyone seeking an identification, yes?
Yeah, absolutely. So when people are seeking an identification, what I really want to see is the cap of the mushroom. Actually, here's a mushroom that a friend crocheted for me that's sitting on my desk. So
That's gorgeous.
Isn't it lovely? But yeah, so I want to see the cap of the mushroom. I also want to see underneath and see what the gills look like, if it has gills. Some mushrooms will have a ring around the stem, which is like a little bit of a skirt. So I want to be able to see that if that's there. Really good to see what it's growing from. And then if you can, cut one in half lengthways through the cap and stem, so kind of that way, and see if it changes colour. But yeah, those are the key features for trying to identify a mushroom.
And don't give any information on taste.
Yeah, I should say, so it is safe to handle mushrooms. Just always make sure you wash your hands afterwards. If you're worried about not being able to wash your hands, just wear gloves. It is safe to handle them, but don't eat them, don't lick them, don't rub your eyes straight after touching them.
Don't sniff them so strongly your nose touches the like don't sniff a phenol in that kind of way or something like that.
Yeah, I mean, I probably wouldn't rub it up inside your nostrils, but you can get pretty close for a sniff, and sometimes you do.
Okay, cool.
Yeah.
Cool. We touched before on the importance of submissions from people involved through Fungi site to let you know where they're spotting these mushrooms. Is that kind of crowdsourced science or crowdsourced material common in fungi? You see it a lot in bird and frog research, for example, but this is the first time I've really seen it done in the mycology field.
I think I might have been the first person doing it oh no, I wasn't. So Fungi Map is another organisation that does something similar. So they're based through the Royal Botanic Gardens, Victoria. And they're interested in a whole lot of other mushroom species, not just, one genus or anything. But I think I was really the first one to take it off, not take it off, the first one to really take it to Facebook and just kind of spread it on social media. Around the same time in New Zealand, they developed iNaturalist and they started using iNaturalist for some mushroom projects. But again, mostly it was plants or animals or whatever. So yeah, I don't think many people bring mushrooms to social media quite as much, but not in the citizen science way.
Maybe they should. It's quite an interesting field. You mentioned, so of course this research study has been published. It was your master's research. You're working on your PhD now?
Yeah, sure am. At the moment, I'm studying the same genus, but not that one specific poisonous species. I'm looking at this one genus broadly, the field mushroom genus. I'm looking to see what's in Australia, how it's related to other species of the same genus in Australia and in other parts of the world. Basically just looking at the evolution of the genus in Australia. Well, Australasia, actually.
Which is quite important because this kind of genus, I can't remember if I'm using genus correctly there or not, is quite common globally, or there's examples of it globally. But in Australia, it's not so well documented.
Yep. Yeah, so that's a problem for fungi in Australia just in general. There's been some historical issues around that. So when white settlers came to Australia, there used to be this belief that all species of fungi are everywhere. And so they believed that the mushrooms they would come across in Australia would be exactly the same as the mushrooms they came across back home.
That must have
And
been a shock.
yeah. It's a similar thing happened to birds, I believe, which is why we've got, magpies in different parts of the world that are totally different. But yeah, so I think something similar happened with birds, but they caught it a bit earlier. With mushrooms, it's 200 years and we're still working it out. So when white settlers came to Australia, they didn't record what they brought with them. They didn't record what was here previously. And then they went around naming everything that they found in Australia as the same species as they found back home, when it's not actually the same species. So it's been a big mess.
And then sorry, please go on.
And then there's the issue of not enough mycologists and not enough funding. So at the moment, I think there are 4 full-time employed mushroom mycologists. So like the fungi that produce mushrooms as opposed to the fungi that produce molds. So there's only 4 of us, I think, in Australia, maybe 5. So there's a lot of work to do and not many people to do it, and just historical issues as well.
So what was it that drew you to mycology? It's such a niche field with a lot of time in the field. What was it that pulled you in?
Honestly, it was an accident. I shouldn't say it like that. It was more of just a chain of events, I guess. I had planned on doing botany, a master's in botany, and the supervisor and I, who planned a project, it just fell through. This project on plants just didn't seem right for me. And at the time, I was really looking around to work out what on earth I would do instead, trying to find another supervisor, trying to find another project. And I'd been doing an internship at the Royal Botanic Gardens. And during that internship, I was working with Teresa LaBelle, a mycologist. And I just got on really well with her. We worked really well together. We had a lot of fun. And she just kept dropping hints like, "Oh, Grace, if you want to do a master's, let me know. I can help you out." And eventually I just took her up on it and said, "Sure, let's do a master's."
It's a great way to fall into it from the sounds of it. A lot of fun. We do have a question coming in from a viewer. Harriet asks, as it's an introduced species, she's curious how widespread. I had it perfectly in my head before we started the stream, and now Agaricus Xanthodermus, thank you, is just wondering how widespread it is throughout Victoria and Australia, and whether fungicide has helped understanding of its distribution as well.
Oh, yeah. Oh, hey, Harriet. Get my words straight and my thoughts in the right order. Yes, Agaricus xanthodermus is very widely distributed throughout Australia, actually. So it's found in Western Australia, South Australia, Victoria, New South Wales, Tasmania, and Queensland. I don't know if it's found in Darwin, but it's basically found everywhere. And it's been interesting learning a bit about the spread of it. So looking back at the first records of when it was observed in South Australia, Victoria, and New South Wales. Anecdotally, it appears to have only started to spread in Victoria in about the 60s or 70s, which is why you might have a lot of parents, aunts and uncles who used to forage growing up, and then they stopped foraging in their lifetime. And the reason they stopped foraging is often because they came across this mushroom instead of the edible mushrooms they were looking for.
So how did it spread out here? Was it just carelessness with soils and stuff like that, or was it something else?
A bit of a mystery. There is a theory that it's to do with the land use. So we find Agaricus xanthodermus, this yellow stainer, really likes urban environments. whereas the edible species that people used to forage for and still do forage for, Agaricus arvensis, which I think is the horse mushroom. Agaricus arvensis tends to like paddocks. So we think it's something to do with land usage, with the urbanization of an area. Agaricus xanthodermis just seems to do a lot better in urban environments. But it could also be people picking mushrooms and walking around with them and spreading the spores, getting the spores on their shoes, finding a bunch, putting it in their wheelie bin because they've gone, oh no, this isn't edible, putting it in their wheelie bin and then wheeling the bin out on bin night and maybe the bin tipping over and dropping the mushrooms on the nature strip, anything like that.
And of course, spreading them to the tip site as well in the process. Thank you so much, Grace, for your time. Just conscious of it. You've given us a lot. What ways can people stay in touch with your research and fungicide as well?
Well, they can submit sightings through iNaturalist. Fungicide has a project on iNaturalist, but if they're wanting to keep in touch with what I'm doing and the outcomes of my research, Facebook or Instagram. Fungicide is on both, and I do regular updates on both of those. Instagram, you'll see a bit more of my day-to-day stuff, whereas Facebook is more like the news posts.
Yeah. And on Instagram, it's at fungi.sight, correct?
That's right, yep.
Very good. Very good. Thank you so much, Grace, for your time, and thank you to everyone for watching and sending in questions, which has been really nice. If you want to catch up with anything from this interview, if you've joined in late and want to catch up, the archive will be uploaded to the MELMAG YouTube account in next half hour or so keep an eye out there. If you want to keep up with MELMAG, we're at MELMAG Vic on every platform, I think now. Just keep in touch. Thanks so much.